Hello, let's discuss on the central dogma about the flow of genetic informations. According to this hypothesis, the genetic information that is being used for synthesizing the protein from the DNA is being first transcribed, that means copied, into the RNA, and this genetic information transcribed in this RNA is being used for synthesizing the amino acids in the proteins. So if this is the DNA, then in order to synthesize this protein, the genetic informations from this DNA will be first transcribed or copied into the mRNA. So this process of copying of genetic information from DNA into mRNA is called as the transcription, and the genetic information that is present in this mRNA will be used for synthesizing proteins. So this process of synthesizing the protein from the genetic information that is stored in this mRNA is called as translation. Now we shall be discussing on the entire mechanisms of this transcription. The mechanism of transcription in prokaryotes involve three steps. The first step is the initiation step. Now, on the signal from the cytoplasm of the cell, the histone proteins that are associated with this DNA will be removed first, and the RNA polymerase will come and bind to the region from where the transcription will first start in the DNA. Now, the RNA polymerase contain a factor called as the sigma factor, which is used for recognizing the region of the DNA from where the transcription will start. So once the sigma factor recognizes from where the transcription will start, the RNA polymerase will come and bind to this region, and it will cause unwinding, that means separation of the two strands of the DNA. So once the two strands of the DNA are being separated or unwinded, the double-stranded DNA, that means DS stand for double-stranded DNA. So the double-stranded DNA will expose its nitrogenous base, and here out of the two strands of the DNA, only one strand will be transcribed and will be used for transcription process. The other strand of the DNA is just a complementary strand. So this the strand of the DNA which carries the genetic information will be used for transcription. So this 3' to 5' strands of the DNA is used for transcription process. So this strand which contain genetic information and is used for transcription is called as the template strand. Now, once the nitrogenous base are exposed due to unwinding of this DNA double helix strand, the ribonucleotide triphosphate, that is the ATP, adenine triphosphate, guanosine triphosphate, cytosine triphosphate, and cytidine triphosphate, and uridine triphosphate. In case of DNA, it will be thymidine triphosphate, but since it is a RNA, it will be uridine triphosphate. So this triphosphate will come and binds to the unwinded, that is the separated strands of the DNA. Now, once this RNA polymerase come and bind to this, it will initiate the formation of mRNA, and the base of this RNA will be paired with the nitrogenous base of the template strand of the DNA. So the base pairing will be on the basis of the base pairing rule. That means A always pair with T, G always pair with C. In case of mRNA, it will be U instead of T. So if the template strand of the DNA have A, it will be T instead of U in the side of RNA. If this side is C, this side will be G. If this side is T, this side will be A. Now this is how the transcription process goes on, as the mRNA cause separation of the two strands of the DNA, and this will cause elongation of the mRNA strands. So this is how the initiations of the transcription process starts. Now the second steps will be elongation. The once initiation starts, various ribonucleotide triphosphate will come and join to the template strand of the DNA. So this ribonucleotide triphosphate will be converted into ribonucleoside monophosphate. That means each phosphate group will be removed. So the pyrophosphate group will go under hydrolysis under the presence of water, and it will form two inorganic phosphate and will be removed together with energy. So this hydrolysis will be initiated by the enzyme pyrophosphatase enzyme. Now the newly formed monophosphate will come and join to the template strands through the help of the RNA polymerase enzyme. Now with the incoming new ribonucleoside monophosphate, base pairing will go on, and these strands, the new strands of the mRNA will keep on joining with the old strand, and this will increase the length of the mRNA. Now the new mRNA and the old mRNA will be joined by the formation of a bond called as the phosphodiester bond. This phosphodiester bond formation between the new and the old mRNA will be carried out by the RNA polymerase enzyme itself. So this bond will initiate the formation of a long polynucleotide mRNA strands. So this is how the formation of an elongated mRNA that transcribed the DNA will take place. Now as transcription keeps going on, the strands where transcription is being completed, the strands of the DNA will again come and join together and form a double helix structure. So this mRNA will keep on unwinding. This RNA polymerase will cause separation of the double-stranded DNA and will initiate transcription. And once the transcription is completed in this strand, it will move into the next strand, and this strand will take its original form again. So this is how elongation of the mRNA, the separation of the DNA, at the same time the formation of double helix DNA takes place. Now the third and the last step in the transcription is the termination. As the transcription proceeds, the hybrid DNA RNA, that means the RNA that is containing the information of both the DNA and RNA together, begin to dissociate, freeing the RNA molecules that is being synthesized. When the RNA polymerase reach to the terminator signal in the DNA, the RNA polymerase terminates, leaving the DNA one after the other, forming a long hybrid chain of the DNA RNA strands. So during the transcription process, it is the RNA polymerase enzyme that catalyzes all the three functions, that is the initiations, elongation, and even termination. Now the newly formed mRNA will contain the genetic information of this transcribed DNA as well. So this mRNA that is containing the genetic information of this DNA will be used for synthesizing of protein. So this is all about transcription.